One of the methods we use to determine the abundance and distribution of fish on Lake George is seining. A seine is a large, long net with a pouch in its center. The seine is pulled through the water, corralling fish in its path, and then pulled on shore. We capture fish to identify the species that are present, as well as their age, length, and their weight. Here is a largemouth bass. Fish biologists lead this sampling. Another method is snorkeling, in which researchers visually identify the fish underwater. This technique works well in areas that are difficult to seine because of obstructions like rocks. Another method of sampling the fish is electrofishing. This is a non-lethal shock that stuns the fish and brings them to the surface where they can be netted and identified. What fish do we find? Long ear sunfish, largemouth bass, bluegill, pumpkin seed, and yellow perch. There are several methods for sampling invertebrate animals in the lake. A weighted grab sampler grabs all mud, debris, and plants from an area. Once on the surface, the animals are sorted out from the mud and plants and then identified. In dip netting, a long net on the end of a pole is dipped into the water to collect invertebrate animals, insects, and mollusks. This dip net sample from Lake George shows an Asian clam, which is an invasive species. What lake invertebrates do we find? Mystery snails, Asian clams, and caddisflies. The Jefferson project is developing a new method for sampling plankton that uses image recognition software to identify pictures of plankton collected with a towable camera. A fine mesh zooplankton net can also be used to collect zooplankton for identification under a microscope. This style of sampler can trap zooplankton at a specific depth. This allows researchers to determine the depths at which different species of zooplankton live throughout the day and night. The three major categories of plankton that we find are Cladocerans, copepods, and rotifers. Samplers are used on the 10 major streams entering Lake George. Using leaf packs containing a known weight of leaves, researchers can measure and compare the rates of leaf decomposition in different streams. Dendy samplers collect invertebrate animals. What stream insects do we find? Damselflies, scuds, mayflies, and finally stoneflies.